Cellular respiration is the set of metabolic reactions and processes that take place in the cells of organisms to convert biochemical energy from nutrients into adenosine triphosphate, and then release waste products. The reactions involved in respiration are catabolic reactions, which break large molecules into smaller ones, releasing energy in the process. As weak so-called high-energy bonds are replaced by stronger bonds in the products. Respiration is one of the key ways a cell gains useful energy to fuel cellular activity. Cellular respiration is considered an exothermic redox reaction which releases heat. The overall reaction occurs in a series of biochemical steps, most of which are redox reactions themselves. Although technically, cellular respiration is a combustion reaction, it clearly does not resemble one when it occurs in a living cell due to slow release of energy from the series of reactions. Nutrients that are commonly used by animal and plant cells in respiration include sugar, amino acids and fatty acids. And the most common oxidizing agent is molecular oxygen. The chemical energy stored in ATP can then be used to drive processes requiring energy, including biosynthesis, locomotion or transportation of molecules across cell membranes. Aerobic respiration. Aerobic respiration requires oxygen in order to generate ATP, although carbohydrates, fats, and proteins are consumed as reactants. It is the preferred method of pyruvate breakdown in glycolysis and requires that pyruvate enter the mitochondria in order to be fully oxidized by the Krebs cycle. The products of this process are carbon dioxide and water, but the energy transferred is used to break strong bonds in ADP as the third phosphate group is added to form ATP. By substrate-level phosphorylation, NADH and FADH2, the negative delta G indicates that the reaction can occur spontaneously. The potential of NADH and FADH2 is converted to more ATP through an electron transport chain with oxygen as the terminal electron acceptor. Most of the ATP produced by aerobic cellular respiration is made by oxidative phosphorylation. This works by the energy released in the consumption of pyruvate being used to create a chemiosmotic potential by pumping protons across a membrane. This potential is then used to drive ATP synthase and produce ATP from ADP and a phosphate group. Biology textbooks often state that 38 ATP molecules can be made per oxidized glucose molecule during cellular respiration. However, this maximum yield is never quite reached due to losses as well as the cost of moving pyruvate and ADP into the mitochondrial matrix, and current estimates range around 29 to 30 ATP per glucose. Aerobic metabolism is up to 15 times more efficient than anaerobic metabolism. However, some anaerobic organisms, such as methanogens, are able to continue with anaerobic respiration, yielding more ATP by using other inorganic molecules as final electron acceptors in the electron transport chain. They share the initial pathway of glycolysis, but aerobic metabolism continues with the Krebs cycle and oxidative phosphorylation. The post-glycolytic reactions take place in the mitochondria in eukaryotic cells, and in the cytoplasm in prokaryotic cells. Glycolysis is a metabolic pathway that takes place in the cytosol of cells in all living organisms. This pathway can function with or without the presence of oxygen. In humans, aerobic conditions produce pyruvate and anaerobic conditions produce lactate. In aerobic conditions, the process converts one molecule of glucose into two molecules of pyruvate, generating energy in the form of two net molecules of ATP. 4 molecules of ATP per glucose are actually produced, however, 2 are consumed as part of the preparatory phase. The initial phosphorylation of glucose is required to increase the reactivity in order for the molecule to be cleaved into two pyruvate molecules by the enzyme aldolase. During the payoff phase of glycolysis, 4 phosphate groups are transferred to ADP by substrate-level phosphorylation to make 4 ATP, and 2 NADH are produced when the pyruvate are oxidized. The overall reaction can be expressed this way: glucose plus 2 Namibian dollars plus 2 pi plus 2 ADP 2 pyruvate plus 2 NADH plus 2 ATP plus 2H plus 2 H2O plus heat. Starting with glucose, 1 ATP is used to donate a phosphate to glucose to produce glucose 6-phosphate. Glycogen can be converted into glucose 6-phosphate as well with the help of glycogen phosphorylase. During energy metabolism, glucose 6-phosphate becomes fructose 6-phosphate. An additional ATP is used to phosphorylate fructose 6-phosphate into fructose 1,6-disphosphate by the help of phosphofructokinase. Fructose 1,6-diphosphate then splits into 2 phosphorylated molecules with 3 carbon chains, which later degrades into pyruvate. Oxidative decarboxylation of pyruvate. Pyruvate is oxidized to acetyl-CoA and CO2 by the pyruvate dehydrogenase complex. The PDC contains multiple copies of 3 enzymes and is located in the mitochondria of eukaryotic cells and in the cytosol of prokaryotes. In the conversion of pyruvate to acetyl-CoA, 1 molecule of NADH and 1 molecule of CO2 is formed. Citric acid cycle. This is also called the Krebs cycle or the tricarboxylic acid cycle. When oxygen is present, acetyl-CoA is produced from the pyruvate molecules created from glycolysis. When oxygen is present, the mitochondria will undergo aerobic respiration, which leads to the Krebs cycle. However, if oxygen is not present, fermentation of the pyruvate molecule will occur. In the presence of oxygen, when acetyl-CoA is produced, the molecule then enters the citric acid cycle inside the mitochondrial matrix and is oxidized to CO2 while at the same time reducing NAD to NADH. NADH can be used by the electron transport chain to create further ATP as part of oxidative phosphorylation. To fully oxidize the equivalent of 1 glucose molecule, 2 acetyl-CoA must be metabolized by the Krebs cycle. 2 waste products, H2O and CO2, are created during this cycle. The citric acid cycle is an 8-step process involving different enzymes and coenzymes. During the cycle, acetyl-CoA plus oxaloacetate yields citrate, which is rearranged to a more reactive form called isocitrate. Isocitrate is modified to become alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and, finally, oxaloacetate. The net gain of high-energy compounds from one cycle is 3 NADH, 1 FADH2, and 1 GTP. The GTP may subsequently be used to produce ATP. Thus, the total yield from one glucose molecule is 6 NADH, 2 FADH2, and 2 ATP. Oxidative phosphorylation in eukaryotes. Oxidative phosphorylation occurs in the mitochondrial crystal. It comprises the electron transport chain that establishes a proton gradient across the boundary of inner membrane by oxidizing the NADH produced from the Krebs cycle. ATP is synthesized by the ATP synthase enzyme when the chemiosmotic gradient is used to drive the phosphorylation of ADP. The electrons are finally transferred to exogenous oxygen and, with the addition of 2 protons, water is formed. Efficiency of ATP production. The table below describes the reactions involved when one glucose molecule is fully oxidized into carbon dioxide. It is assumed that all the reduced coenzymes are oxidized by the electron transport chain and used for oxidative phosphorylation. Although there is a theoretical yield of 38 ATP molecules per glucose during cellular respiration, Such conditions are generally not realized due to losses such as the cost of moving pyruvate, phosphate, and ADP into the mitochondria. All are actively transported using carriers that utilize the stored energy in the proton electrochemical gradient. Pyruvate is taken up by a specific, low km transporter to bring it into the mitochondrial matrix for oxidation by the pyruvate dehydrogenase complex. The phosphate carrier mediates the electron-neutral exchange of phosphate for O- or symport of phosphate and protons across the inner membrane, and the driving force for moving phosphate ions into the mitochondria is the proton motive force. The ATP-ADP translocase is an antiporter and exchanges ADP and ATP across the inner membrane. The driving force is due to the ATP having a more negative charge than the ADP, and thus it dissipates some of the electrical component of the proton electrochemical gradient. The outcome of these transport processes using the proton electrochemical gradient is that more than 3H+ are needed to make 1 ATP. Obviously, this reduces the theoretical efficiency of the whole process, and the likely maximum is closer to 2,830 ATP molecules. In practice, the efficiency may be even lower due to the inner membrane of the mitochondria being slightly leaky to protons. Other factors may also dissipate the proton gradient, creating an apparently leaky mitochondria. An uncoupling protein known as thermogenin is expressed in some cell types and is a channel that can transport protons. When this protein is active in the inner membrane, it short-circuits the coupling between the electron transport chain and ATP synthesis. The potential energy from the proton gradient is not used to make ATP but generates heat. This is particularly important in brown fat thermogenesis of newborn and hibernating mammals. According to some of newer sources, the ATP yield during aerobic respiration is not 36 to 38, but only about 30-32 ATP molecules, 1 molecule of glucose, because ATP, NADH plus H plus and ATP, FADH2 ratios during the oxidative phosphorylation appear to be not 3 and 2, but 2.5 and 1.5 respectively. Unlike in the substrate-level phosphorylation, the stoichiometry here is difficult to establish. ATP synthase produces 1 ATP, 3H+. However, the exchange of matrix ATP for cytosolic ADP and pi mediated by ATP-ADP translocase and phosphate carrier consumes 1H+, 1 ATP due to regeneration of the transmembrane potential change during this transfer. So the net ratio is 1 ATP, 4H+. The mitochondrial electron transport chain proton pump transfers across the inner membrane 10H+, 1 NADH plus H+, or 6H+, 1 FADH2. So the final stoichiometry is 1 NADH plus H+, 10H+, 10 quarters ATP equals 1 NADH plus H+, 2.5 ATP 1 FADH2. 6H+, 6 quarters ATP equals 1 FADH2, 1.5 ATP NADH plus H plus coming from glycolysis ratio during the oxidative phosphorylation is 1.5, as for FADH2. If hydrogen atoms are transferred from cytosolic NADH plus H plus to mitochondrial fat by the glycerol phosphate shuttle located in the inner mitochondrial membrane, 2.5 in case of malate aspartate shuttle transferring hydrogen atoms from cytosolic NADH plus H plus to mitochondrial NAD plus. So finally we have, per molecule of glucose substrate-level phosphorylation, 2 ATP from glycolysis plus 2 ATP from Krebs cycle. Oxidative phosphorylation 2 NADH plus H plus from glycolysis. 2 times 1.5 ATP or 2 times 2.5 ATP to NADH plus H plus from the oxidative decarboxylation of pyruvate, and 6 from Krebs cycle. A times 2.5 ATP to FADH2 from the Krebs cycle. 2 times 1.5 ATP. Altogether, this gives 4 plus 3 plus 20 plus 3 equals 30 ATP per molecule of glucose. The total ATP yield in ethanol or lactic acid fermentation is only 2 molecules coming from glycolysis, because pyruvate is not transferred to the mitochondrion and finally oxidized to the carbon dioxide, but reduced to ethanol or lactic acid in the cytoplasm. Fermentation. Without oxygen, pyruvate is not metabolized by cellular respiration but undergoes a process of fermentation. The pyruvate is not transported into the mitochondrion, but remains in the cytoplasm, where it is converted to waste products that may be removed from the cell. This serves the purpose of oxidizing the electron carriers so that they can perform glycolysis again and removing the excess pyruvate. Fermentation oxidizes NADH to NAD+, so it can be reused in glycolysis. In the absence of oxygen, fermentation prevents the buildup of NADH in the cytoplasm and provides NAD+ for glycolysis. This waste product varies depending on the organism. In skeletal muscles, the waste product is lactic acid. This type of fermentation is called lactic acid fermentation. In strenuous exercise, when energy demands exceed energy supply, the respiratory chain cannot process all of the hydrogen atoms joined by NADH. During anaerobic glycolysis, NAD+ regenerates when pairs of hydrogen combine with pyruvate to form lactate. Lactate formation is catalyzed by lactate dehydrogenase in a reversible reaction. Lactate can also be used as an indirect precursor for liver glycogen. During recovery, when oxygen becomes available, NAD+ attaches to hydrogen from lactate to form ATP. In yeast, the waste products are ethanol and carbon dioxide. This type of fermentation is known as alcoholic or ethanol fermentation. The ATP generated in this process is made by substrate-level phosphorylation, which does not require oxygen. Fermentation is less efficient at using the energy from glucose. Only 2 ATP are produced per glucose, compared to the 38 ATP per glucose nominally produced by aerobic respiration. This is because the waste products of fermentation still contain chemical potential energy that can be released by oxidation. Ethanol, for example, can be burned in an internal combustion engine like gasoline. Glycolytic ATP, however, is created more quickly. For prokaryotes to continue a rapid growth rate when they are shifted from an aerobic environment to an anaerobic environment, they must increase the rate of the glycolytic reactions. For multicellular organisms, during short bursts of strenuous activity, muscle cells use fermentation to supplement the ATP production from the slower aerobic respiration. So fermentation may be used by a cell even before the oxygen levels are depleted, as is the case in sports that do not require athletes to pace themselves, such as sprinting. Anaerobic respiration. Cellular respiration is the process by which biological fuels are oxidized in the presence of an inorganic electron acceptor to produce large amounts of energy. To drive the bulk production of ATP, anaerobic respiration is used by some microorganisms in which neither oxygen nor pyruvate derivatives as the final electron acceptor. Rather, an inorganic acceptor such as sulfate or nitrate is used. Such organisms are typically found in unusual places such as underwater caves or near the lava chutes at the bottom of the ocean.